so this is my second video, part 2 on AP Bio topic 2.8. My last video cut me off, so here we go. We're going to focus on different examples of osmoregulation, or how organisms can control or maintain water balance so that their internal environment is, in animals, isotonic to their external environment. So let's start with a unicellular eukaryotic organism called a paramecium. So this paramecium, as well as like amoebas and other unicellular protists, if they live in fresh water, they are living in a hypotonic environment. So they have this constant battle of water flowing from the hypotonic external environment inside of their cell. And so with that, they run the risk, if they don't osmoregulate and pump this extra water out, they run the risk of swelling and expanding like we saw in animal cells, and potentially bursting and undergoing cell lysis. So thankfully, these little organisms have this special organelle. I just zoomed in. So there it is. It's called a contractile vacuole. So if we zoom in and take a look at this contractile vacuole, this is the organelle that they use to osmoregulate and to pump excess water out of their cell, of them. And so let's go ahead and see how a contractile vacuole works. So in the lipid bilayer of the contractile vacuole, there are protein pumps. Now, I use the word pump because these proteins are going to use active transport. So this is earlier in Unit 2. So they're going to be pumping ions, in this case hydrogen ions, against the concentration gradient from a low concentration outside in their cytoplasm. Remember, this is all happening within a paramecium. So going from a lower concentration to a higher concentration. So by using active transport for this, they are creating a hypertonic space within that contractile vacuole. Now, why this is so rad is because now water molecules are going to follow by osmosis. So they're like hypotonic outside of this contractile vacuole, so in the cytoplasm. Now they're going to flow within that contractile vacuole by osmosis, passive transport. Now this causes that contractile vacuole to swell and gain mass, right? It's gaining water volume. So the contractile vacuole increases in size. Now, the little paramecium, so keep in mind, it's in a hypotonic freshwater environment. So water is flowing through its cell membrane into the cytoplasm. Then from there, the contractile vacuole organelle creates a hypertonic environment that now water is going to flow from the cytoplasm into this contractile vacuole. This contractile vacuole is then going to use ATP, there's the words, going to use ATP to cause it to now contract, that's where the name comes from, and pump that excess water out. And this is how, and this repeats over and over and over. So this is how that cute little paramecium, how it osmoregulates. And so if we look again, there are those contractile vacuoles, and that is an example of one way organisms osmoregulate. Now, let's go ahead and talk about fish who literally live in water, right? So we have freshwater fish and we have saltwater fish. So a freshwater fish's challenge is going to be that it lives in a hypotonic environment. Now, animal cells prefer to be isotonic. So they need equal solute concentrations both inside and outside of the cells. Now, a freshwater fish, not only does it have water diffusing just from its environment through its cells in its body, but it also, when it eats, it's taking in large amounts of fresh hypotonic water. So how does it handle this, right? So if you're taking in fresh water, you need to get rid of it, but also you got to figure out a way to add ions. So in the gills, where the fish does gas exchange and, has oxygen diffuse in from the water and carbon dioxide diffuse out, there's actually also going to be active transport pumps, ion pumps, and the fish are actually going to use active transport to pump ions into their bloodstream to counterbalance that fresh water that is being taken in when it eats. The second thing these freshwater fish do is they're going to have large amounts of dilute urine. They're going to just pee out a lot of this excess water. Think about the last time you as a human drank a ton of water. You probably went to the bathroom quite a bit. And that's kind of what freshwater fish do as well. Now, saltwater fish, though, has the opposite challenge, right? So they are like, anytime they eat, they're taking in tons of ions, and they're living in a hypertonic environment. Lots of solutes externally. So what they do, it's the opposite. Their gills have active transport ion pumps to pump ions out. There we go. To pump ions out. And if we think about the urine, instead of wasting water through their urine, instead, they're going to have very little urine, and it's going to be highly concentrated. Now, another, just a quick mention about fish is if you have like goldfish or betta fish, like in a bowl without a filter, you might think, okay, they're freshwater fish. However, they're going to the bathroom all the time in that water, and so that's nitrogen waste. So the wastes, metabolic wastes it's called, can actually accumulate in the water, creating a hypertonic environment. And if you don't filter that water, either by putting a plant where roots can take the nitrogen out or an actual filter, the freshwater fish bowl will become hypertonic, and it will result in your fish dying. So change the water or get a plant or a filter. All right, so their urine, very little water leaves in their concentrated urine. And then I believe my last topic on osmoregulation is humans. So we function best with isotonic conditions, and it is actually going to be our kidneys that work really hard to maintain our blood osmolarity, to keep our blood solute concentration isotonic with our blood cells. So we actually use hormones for this. There's a particular hormone called ADH, antidiuretic hormone, that if you're in AP Bio, we'll talk more about in Unit 4 on negative feedback. But anyway, let's go ahead and take a quick look at our kidneys. So this is my last slide, and just, I know it's not animated, but a quick diagram to see. So if a person is maybe like a little dehydrated, they're not really drinking a lot of water, what the hormone ADH does when it travels through our bloodstream and reaches the target cells in our kidneys, there's a region called the collecting duct that as our blood is filtered and it creates filtrate, which becomes our urine, there's a tube called the collecting duct that is where that filtrate travels to our bladder. So if you are dehydrated, we don't want to pee out water and waste it. That would further dehydrate us, creating an even more hypertonic solution around our cells. So what happens is ADH is going to communicate with the target cells of the collecting duct to actually increase the amount of aquaporins in that collecting duct. Now what that does is as the filtrate, or what will become your urine, travels through that collecting duct, water is going to diffuse out by osmosis through those aquaporins into the tissues of your kidneys, therefore conserving water. I should point out that if you look at this diagram, the medulla region of your kidneys is actually very hypertonic. This color, like this ombre color scheme where you can see in the top of this diagram the light peach color, and then it gets darker as you go down, that's like showing it gets more and more hypertonic. So here the water is going to diffuse out by osmosis to conserve water. Now on the opposite, let's say that you were overhydrated. You have drank water all day long. You are not going to release ADH. ADH stands for antidiuretic hormone. It's released to conserve water. So if you are overhydrated and you have a hypotonic blood, basically, or body tissues, what will happen is you will actually decrease the amount of aquaporins in that collecting duct. And you can see in this diagram that allows for lots of water to enter into your bladder to be expelled from your body, therefore helping us to osmoregulate and maintain isotonic conditions. So our blood osmolarity is based under like hormonal control. Okay, so that is it for my few examples of osmoregulation, and I hope it was helpful. Great job.